Have you ever stopped to think about how a tiny little pill can have such a massive impact on your body? I mean, how does it know exactly where to go and what to do once it's inside you? Well, it's not magic, it's a fascinating science called pharmacodynamics. And today, we're going to pull back the curtain and see exactly how it all works. So here's the game plan. We're going to follow a drug's journey through 6 key steps. First, we'll get on the same page about what we're even talking about. Then we'll witness its very first handshake with the body, explore the different kinds of messages it can send, and see how it rings the cell's doorbell to get that message inside. And finally, we'll figure out how we measure a drug's power and talk about the all-important margin of safety between helping and harming.
All right, let's dive right in at the beginning.
What are we really asking here? Well, think about it. You get a headache, you pop a pill, and somehow that pill navigates the chaos of your body to target the pain. It really does feel like magic, doesn't it? But it's pure science, and that science has a name.
And that science is pharmacodynamics. In simple terms, it's the study of what a drug does to the body. It's the whole story, the entire mechanism of action, from that very first point of contact all the way to the final effect that we actually feel.
So where does this whole process kick off? Well, a drug can't just wander in and start giving orders. It first needs to find its specific partner, its designated target inside the body. This is that first crucial handshake.
You can think of drugs like highly specialized messengers. They've got a very specific address written on them. They don't just float around randomly, they are designed to seek out and bind to very specific targets. And the 2 most common targets on their list are enzymes and receptors. Let's tackle enzymes first.
Okay, so enzymes are basically the catalysts that speed up reactions in our body. Drugs can block or inhibit them in 2 main ways. A competitive inhibitor is like someone stealing your designated parking spot. It binds right to the enzyme's main active site, physically blocking the normal molecule that's supposed to park there. A non-competitive inhibitor, though, is a bit sneakier. It binds to a totally different spot, changing the enzyme's shape. It's like someone putting a boot on your car. The parking spot is still there, but it's now useless. The key difference, as you can see, is that you can overcome competitive inhibition by just flooding the area with more of the normal molecule. With non-competitive inhibition, no chance.
Now, I know graphs like this can look a little intense, but it's actually a fantastic visual for telling these inhibitors apart. Just follow the lines. The black line is our enzyme working at its normal speed. Now, see the red line for competitive inhibition? It's slowed down for sure, but notice how it eventually reaches the same maximum speed, the same height? But the blue line for our non-competitive inhibitor, its peak is way lower. Its maximum speed has been permanently nerfed. It's a really clear visual fingerprint.
Okay, so that's enzymes. But what about that other major target, receptors? This is where the story gets really fun, and we can use one of the best analogies in all of biology, a lock and a key.
To get this, we just need 2 key ideas. First is affinity. That's simply how well the drug key fits into the receptor lock. A perfect fit means high affinity. But just fitting isn't enough, right? That brings us to the second term, intrinsic activity. This is the big one. Can the key actually turn the lock and open the door? To do anything, a drug needs both. It has to fit and it has to turn.
And this chart shows us the different kinds of keys we can have. An agonist is the master key. It fits perfectly, turns the lock all the way, and gives you a full 100% effect. A partial agonist is like a poorly cut key. It fits, sure, but it only turns the lock part way, so you get a weaker response. And then you've got the antagonist. This is a fake key. It slides into the lock perfectly, blocking the real key from getting in, but it can't turn at all. It has affinity, but absolutely zero intrinsic activity. It's just a blocker.
So the drug has bound to the receptor on the outside of the cell. The key is in the lock, and it's turned. But now what? The message is still on the outside. How does the signal get from the outside of the cell to the inside? How does it actually ring the cellular doorbell?
Well, cells have several different kinds of doorbells. Some are super fast ion channels, others are linked directly to enzymes, and some drugs even bypass the door and go straight inside the cell. But one of the most common and important types, the target for a huge number of modern drugs, are the G-protein coupled receptors, or GPCRs for short.
Here's the bird's eye view. The drug comes along and binds to this big receptor that's snaking its way through the cell membrane. That binding action is the finger pressing the doorbell, and that press causes a change on the inside of the cell, which wakes up something called a G protein.
Now, if we zoom in on the wiring, we see the G protein actually has 3 little parts. When the doorbell rings, the alpha part gets a jolt of energy. It breaks away and acts like a little runner, carrying the message deeper into the cell. This runner then goes and activates other processes, setting off this whole chain reaction, or cascade, that produces the drug's final effect. It's just this elegant relay race of signals.
We've been way down in the microscopic details. Now let's zoom all the way back out. In the real world, how do we actually compare different drugs? How do we measure their overall power?
To measure a drug's power, we have to get 2 very important terms straight: potency and efficacy. Potency is all about the amount of drug you need. If you only need a tiny bit to get an effect, that drug is very potent. Efficacy, on the other hand, is about the maximum effect a drug can produce. It's the ceiling of its power, no matter how high the dose goes. And this graph makes the difference crystal clear. Potency is measured left to right. The further to the left a drug's curve is, the more potent it is, because you need less of it. So right here, drug D is our winner for potency, but efficacy is measured from bottom to top. It's all about how high the curve's peak is. And here, drug C has the greatest maximum effect, making it the most efficacious. And here's the big takeaway: the most potent drug is not always the most effective one. This brings us to our final, and maybe the most important question of all. A drug can be potent, it can be effective, but is it safe? We have to know the distance between a dose that helps and a dose that could harm. This is how we visualize a drug's safety profile. We've got 2 curves. The one on the left shows the effective dose. Its midpoint, the ED50, is the dose that's effective for half the population. The curve on the right represents the toxic or lethal dose. Its midpoint, the LD50, is the dose that's lethal in 50% of animal tests. That space between these 2 curves, that is the all-important margin of safety. And we can actually put a number on that safety gap using this simple formula, the therapeutic index. You just divide the lethal dose, LD50, by the effective dose, ED50. The bigger the final number, the wider the margin of safety, and the safer the drug is considered to be. It's a simple but incredibly powerful calculation in medicine.
And so that's our journey, from a drug's first molecular handshake to the cascade of signals it unleashes inside our cells. It's all a masterpiece of biochemical engineering. But it always comes down to this fundamental trade-off. For every medicine, the goal is to make that gap between help and harm as wide as possible. Which leaves us with the really tough question that scientists and doctors face every single day: how wide is wide enough?